Here's a question that shows up constantly on exam blocks. What does cyclosporine actually shut down? It's IL-2, the single main growth factor for T cells, and the same molecule doctors give at high dose to fight cancer. Everything in cell-mediated immunity, from macrophage activation to killer T cells to natural killer cells, traces back to this one signaling axis. In this video, we'll cover the Th1 response, macrophage activation, cytotoxic T lymphocytes, NK cells and ADCC, and T-cell migration and memory, and I'll flag the timestamps in the description below if you want to jump straight to one. Let's start with the cell that runs the whole show, the Th1 helper T cell. The Th1 subset of helper T cells is the central driver of cell-mediated immunity, and it directs three other players at once. It activates macrophages, CD8, positive cytotoxic T lymphocytes, and natural killer cells. This all gets switched on by two polarizing cytokines, IL-12, coming from macrophages and dendritic cells, and IFN-γ, coming from T cells and NK cells, and together they push naive T cells down the Th1 path and amplify the whole response. So keep this box in your head as your anchor for the whole topic. IL-2 drives T cell growth. IFN-γ activates macrophages and switches B cells to IgG. TNF-alpha drives inflammation and apoptosis, and IL-12 drives Th1 polarization itself. Building on that cytokine axis, the Th1 response is really working toward two goals at once, and the first is activating infected macrophages so they can finish off the pathogens they've already ingested. The second goal is generating cytotoxic T lymphocytes that recognize and eliminate any cell displaying foreign peptides on MHC class I. With those two goals in mind, let's zoom into the first one. What actually happens inside an activated macrophage? Now that we've covered the two goals, let's talk about what classical or M1 activation actually does to a macrophage. IFN-γ is the trigger, and it dramatically boosts the macrophage's microbicidal capacity. As a result, you get increased nitric oxide, increased reactive oxygen intermediates, and increased TNF-alpha, three separate weapons pointed at the ingested pathogen. But activation does something else too. It upregulates MHC class II and co-stimulatory molecules, which is exactly what lets this macrophage turn around and present antigen to a naive Th cell. In other words, that antigen presentation from the last scene is really a three-signal handshake. Signal 1 is the T-cell receptor engaging the MHC class II peptide complex on the macrophage. Signal 2 is co-stimulation. Think CD40 binding CD40 ligand and B7 binding CD28. Signal 3 is cytokines IL-12 and IFN-γ, and together these three signals are what drive Th1 polarization and clonal expansion. Once that's done, the effector Th1 cells migrate back out to the infected tissue and activate macrophages there through IFN-γ and CD40 ligation, completing the loop. Let's lock this in as a table because it's a favorite exam format. Nitric oxide is microbicidal and works by inhibiting the pathogen's own metabolism. Reactive oxygen species drive the oxidative burst and phagolysosomal killing. TNF-alpha is pro-inflammatory and activates the endothelium along with other immune cells. And MHC class II with co-stimulation is the row that connects effector function back to antigen presentation and T-cell activation, the bridge between the macrophage's killing job and its signaling job. With macrophage effector function covered, there's one more piece of Th1 collaboration worth knowing. IFN-γ from Th1 cells also drives B cells to class switch, specifically toward IgG1 and IgG3 in humans. These IgG antibodies opsonize pathogens, coating them so they're easier to grab. Which is why this matters clinically. Opsonized pathogens get taken up by macrophages through Fc receptors, so this single cytokine is quietly linking your humoral and cellular immune responses together. Now, let's shift from the macrophage side of Th1 immunity to its other major arm, the cytotoxic T lymphocyte. Now that we've moved into the CTL story, the core rule is this. CTLs recognize MHC class I peptide complexes, and because MHC class I presents peptides that come from inside the cell's own cytosol, it's really a window into what's happening inside that cell. As a result, Any cell displaying viral peptides, tumor peptides, or even a foreign allogeneic MHC molecule becomes a target. The exam-critical takeaway is simple. Any cell showing a non-self peptide on MHC class I is fair game for a CTL. Because of that rule, transplant rejection follows directly. HLA is extremely polymorphic between individuals. So a donor's allogeneic MHC molecules simply look foreign to the recipient's CTLs. Which is why you can get a vigorous CTL response against a transplanted organ, even though there's no infection anywhere in the picture. The mismatched MHC alone is enough to trigger it. With that danger case established, let's look at what a CTL needs before it can actually go kill. CTLs can actually be primed by dendritic cells through a process called cross-presentation. But priming alone isn't enough. Optimal CTL expansion requires IL-2, and that IL-2 comes from Th1 cells, tying this whole pathway right back to the Th1 axis we started with. On top of that, IFN-γ and other inflammatory signals upregulate MHC class I on the target cells themselves, which makes those targets even more susceptible to being lysed. Now that the CTL is primed, expanded, and its targets are lit up, let's look at exactly how it kills. So how does a CTL actually deliver the kill? The first route is the Perforin/granzyme pathway. Perforin pours straight through the target cell's membrane. Granzymes, which are serine proteases, slip through those pores and activate caspases, driving the target into apoptosis. The second route is FAS binding FasL. FasL on the CTL surface binds FAS on the target, and that also triggers caspase activation and apoptosis downstream. There's a third, softer route too, cytokine-mediated killing, where IFN-γ and TNF-alpha or TNF-beta can induce apoptosis in sensitive cells on their own. And here's the clinical pearl worth remembering. CTL killing is highly specific and does not damage bystander cells, and the FAS pathway in particular matters for immune privilege and for clearing out activated lymphocytes once they're no longer needed. Quick check before we move on. Based on what we just covered, which molecule on a CTL's surface binds to FAS on the target cell to trigger caspase activation and apoptosis? Drop your answer in the comments below. Let's shift gears now to natural killer cells, which sit on the innate side of the story. They deliver rapid cytotoxicity without needing any prior sensitization. Their activity is set by a balance between activating and inhibitory signals, and the inhibitory receptors specifically recognize MHC class I through HLA. Because of this, loss of MHC class I, which happens commonly in viral infection or in tumors, removes that inhibitory brake and triggers NK killing. It's almost the mirror image of how CTLs work. CTLs need to see non-self on MHCI, while NK cells attack when MHCI disappears altogether. As a result, NK cells carry a specific set of markers worth putting in table form. CD16, also called FGI, binds the Fc portion of IgG and mediates ADCC. We'll come back to that in a moment. CD56, or NCM, handles adhesion and also splits NK cells into bright and dim subsets. NKIRs along with NKG2A are the inhibitory receptors that read MHC class I on other cells, the same brake system we just described. With those markers in mind, NK cells kill through mechanisms that should sound familiar, perforin and granzyme, the exact same porin protease system CTLs use. They can also use FasL. And on top of direct killing, they release cytokines too, specifically IFN-γ and TNF-alpha, reinforcing the same inflammatory signals we saw driving macrophage activation earlier. Now, let's look at the one NK function that bridges all the way back to antibodies, ADCC. Now that we understand CD16, we can talk about ADCC, Antibody-Dependent Cell-Mediated Cytotoxicity, which literally bridges your humoral and cellular immune responses. The effector cells here aren't just NK cells. Macrophages, neutrophils, and eosinophils can all do this too, because they all express CD16 or FGI. When IgG antibodies coat a target, say a virus-infected cell or a tumor cell, That Fc region gets recognized by CD16, and that triggers cytotoxic granule release and lysis of the target. Here's the exam-critical distinction. In ADCC, the specificity is provided by the antibody, not by the effector cell itself. The effector cell is just muscle. The antibody is the aim. Because specificity comes from the antibody, this mechanism gets exploited therapeutically. Monoclonal antibodies like rituximab and trastuzumab rely on Fc engagement as part of how they work. And on the natural infection side, eosinophil-mediated ADCC is a key defense against helminths. One mechanism, two very different exam contexts. With NK cells and ADCC covered, let's zoom out and look at where all these activated cells actually go. Naive T cells get activated in secondary lymphoid organs, lymph nodes, and spleen after they encounter antigen presented by dendritic cells or macrophages. From there, IL-2 takes over as the primary growth factor, acting in both an autocrine and paracrine way to drive clonal proliferation, the same IL-2 we flagged as your exam anchor back at the start of this video. That expansion produces two outputs: effector cells ready to fight now and memory cells ready to respond faster next time. Once they're made, effector lymphocytes leave the lymphoid tissue through the efferent lymphatics and the bloodstream, homing to wherever the infection actually is. Memory cells, meanwhile, stick around long-term and can be reactivated rapidly if you're re-exposed. And here's a connection worth locking in: delayed type hypersensitivity, or DTH, runs on that exact same Th1, macrophage, and CTL machinery. Classic examples are the tuberculin skin test and contact dermatitis from poison ivy, a normal protective response that can also cause tissue damage if it's dysregulated. Now, let's pull everything together with the highest yield facts from this entire topic. Let's close this out with the facts most likely to be tested directly. First, IL-2 is the main T-cell growth factor, and it's the actual drug target of immunosuppressants like cyclosporine and tacrolimus, while high-dose IL-2 itself is used in cancer immunotherapy. Second, IFN-γ activates macrophages and boosts MHC expression, so a deficiency in it leaves you susceptible to intracellular bacteria like mycobacteria and salmonella. Third, Perforin or granzyme deficiency, seen in familial hemophagocytic lymphohistiocytosis, results in defective CTL and NK killing. Fourth, FAS or FasL mutations cause autoimmune lymphoproliferative syndrome, or ELPS, because lymphocytes can no longer undergo apoptosis properly. Fifth, remember that NK cells belong to the innate response, but their activity gets enhanced by Th1 cytokines, which is why they're such a critical early defense against viruses and tumors. And finally, ADCC is exploited therapeutically. Monoclonal antibodies like rituximab and trastuzumab depend on Fc engagement for part of their effect. That's the complete picture of cell-mediated immunity, from the Th1 cytokine axis all the way through to memory. Everything you saw today loops back to one starting point: the Th1 cell, driven by IL-2, orchestrating macrophages, cytotoxic T lymphocytes, and NK cells into one coordinated, highly specific attack on infected or abnormal cells. If this helped, drop your knowledge check answer in the comments, and I'll see you in the next video. Thanks for watching.